welcome to the 2023 Bioanalysis Zone panel discussion on regulations for cell and gene therapies. I'm Ellen Williams, digital editor at Bioanalysis Zone, and I'm here today with my three panelists who would like to introduce themselves. So Eric, should we go with you first?
Certainly. My name is Eric Feely. I'm a senior field application scientist with Gyros Protein Technologies.
My name is Xiaobin Zhang, and I'm a principal scientist in the bioanalysis science and the immunogenicity group at Takeda.
I'm Kimberley Buytaert-Hoefen. I'm the executive director and global head of regulatory affairs at QPS.
Fab. So to kind of kick us off, all off then, what are we talking about when we talk about cell and gene therapies, and how are they currently being used within the bioanalytical space?
Okay, so I'll take a first stab at that. So for our cell and gene therapy workflows, it's pretty broad. It encompasses a lot of different methodologies and designs for treating a lot of disease. So we have cell therapies, which are mostly focused on things like CAR T, which is a useful tool that's been used a lot in cancer. And then we have a lot of workflows now in gene therapies where we have the opportunity to treat a lot of rare diseases and expand beyond that into some modalities that are not currently being well served by other biologics and whatnot.
And it's an amazing opportunity in the industry to move from treating a disease to actually have the potential to cure a disease. And then it's about genetically altering the body, either inside of the body or taking the cells out of the body, and being able to treat something inside of the body. So it's a really exciting time, and we finally made it there commercially.
Compared to the traditional molecular therapy method, the gene therapy and cell therapy, they actually use the genetic material and to deliver this targeting gene into the patient and use it for the disease therapy or for prevent of any disease.
Yeah. Well, exactly like you touched on, this is maybe not a super new modality necessarily, but maybe more newer than some of the more traditional methods. So what are the benefits of using them, say, over some of those maybe small molecule, more traditional methods?
Yes, I think Kim started with the potential to cure disease is really what we're driving at here, where we can make a modification to, some really debilitating diseases, and we can cure it on a gene level. It's a single dosage, it's a single treatment usually for a lot of these gene therapy products, and they're lifelong. So that has the ability to, dramatically change somebody's life with a single treatment, which we can't do with biologic, which requires continuous dosage and things like that.
Yeah, because the traditional molecular you need to multi-dose mainly is try to cure the symptom, not from the root cause, but the gene therapy actually from the root. From the gene, use the gene to deliver the protein and then express the protein or prevent any faulty proteins expressed in the patient.
So what we don't know is what are the long-term effects of these treatments. We know that clinically we're seeing that early efficacy. It's very exciting for patients that have no treatment options. So the regulatory agencies are willing to take that risk, giving them that treatment option, showing that signs of clinical efficacy. But what we have to do is monitor long-term. So we have to see there's shedding studies are important. Is anything getting out into the environment? Reproductive studies will be important. So long-term studies will continue as these products are already out there for the patients.
So you touched a little bit there on some of the limitations. We don't have those long-term studies. So what other limitations are we kind of finding within these modalities?
Well, many of these modalities are for treatments of rare diseases, and we have to truly understand the disease, what's the genetic defect to be able to treat it. But then we also have to know what are the biomarkers that you'll be able to look at over time to show that you're having the efficacy of the product.
One of the other things that we've certainly seen the field transition from, at least in gene therapy, is away from using lentiviral vectors, which have the potential to integrate into the host DNA and could potentially be problematic given the fact that we don't have as much control over where that integration may happen. So there's a lot of concern that could be cancer causing. So as the patient safety is of critical importance, they've mostly utilized lentiviral vectors now for things like cell therapies. But in gene therapy, we've really seen the growth and expansion of AAVs as a modality because it seems to show great promise and seems to be much less dangerous to the patient.
Bioanalytical actually play an important role in this area because we need to monitor where the gene goes and how much. And also we need to monitor what's the patient reaction to this gene therapy product.
Great. And also, route of administration is really difficult. CAR T was an excellent opportunity for blood cancers. You're going around in the blood. Now they're having that be able to move to the solid tumors because of the blood supply to the solid tumors. The eye is a great route of administration, put it right there, but a lot of the other ones are very difficult to get to. The brain, sometimes they use cannulas, and then it requires specific ways to target exactly what you're trying to get to. So that's a huge difficulty in the field.
Thank you.
One other thing on that is some of the AAVs do show some tropism, but I think there's still a lot of work to be done in the field is to show, even within that, how good that tropism is and how tissue specific are those AAV serotypes for getting to the target destination, because some of them are a little broader, I think, than we want them to be.
I was surprised with viral vectors how you can already have exposure to it and you may not be able to get the treatment.
Yeah, that's maybe because of the patient maybe got some immune reactions to this therapy. They deliver the gene to the patient, but the patient's immune system have immune reactions. They may generate antibody against it and get it cleared, and then lost the therapy efficacy.
Yeah, the immunogenicity is a huge topic.
Yeah.
Given the fact that in cases where we have those pre-existing antibodies, we may have to dose at significantly higher amounts than we wanted to, and then we have to a whole nother safety consideration for can we dose at that level, right? And so it's an ongoing escalation that we don't really want to do, but given some of the pre-existing antibodies, we may have no choice.
And you can only have one type of treatment. If you were dealt a bad hand of cards, you couldn't have another gene therapy with that type of viral vector.
Right.
That's so interesting.
Yeah, to some extent, if you increase the dose, sometimes it will prevent the pass through the immune reactions, but sometimes it will not, because if you get higher dose, the immune system will be more stronger, and then lead to more strong side effect, which will hurt the patient. That's why you have to keep monitoring what the immune reaction it is and how much get in.
Yep.
And then I also that was interesting, the samples. You have to have the samples because they're not stable for a long amount of time. So that it's important to be able to get the clinical samples, the preclinical samples to the laboratory maybe within 24, 48 hours?
No, that's unnecessary, because for most of the assays, the protein reaction or the kind of is quite stable. They can be freeze down and then freeze on dry ice, stored at -80 for a longer time. So actually, that's why for most of the assay, the analysis do not need to do in a short time. But for the cell-based assay, because the cell is a living cell, so that means the cellular immune reaction, you have to do it in a short time. That's why in a lot of publications, they can say that for the PBMC, the cellular immune reactions, they have to treat the cells in 8 hours or 10 hours. That's the limit. Yeah,
that's a challenge.
Yeah, challenging part.
Especially considering some of these patient cohorts may be widely disseminated, and so the ability to get to certain patients can be challenging.
Great, thank you. So if we move on a little bit towards the platforms that are currently being used, what are we seeing now being used, and what do you think maybe within the next 5 to 10 years do you think are going to be some of those platforms that are used a bit more regularly within this space?
Actually, in the bioanalysis field for this cell therapy or gene therapy, they need to monitor at the different levels. First, is at the vector delivery vehicles, such as virus. We need to check what the virus level it is, and that's mainly used PCR, qPCR, ddPCR. That's the digital PCR. That's the main tools used to monitor the viral vectors, and not the gene. Because the targeting gene, how much it goes in and where they go. That's they also use the PCR method. And the third is that once the gene going to the patient, is try to detect the proteins. Because once they deliver the gene, the targeting is get the gene expressed in the patient. That is mainly use the ELISA method, like the binding or flow cytometry, to make sure that the targeting gene is expressed on the cells and get the functions. And the fourth part is for the immune reaction of the patient. That's they monitor how the patient reacting to this gene therapy product. That's the four main tools used to monitor in the gene therapy and the cell therapy.
Yeah, and they're just so different, gene and cell therapy. And then within it, everything's different. CAR-T is different from stem cells, which is different from mesenchymal cells, which is different from gene therapy. So, and then how do you do that? PK, PD, I think it's all unique and it all needs to be customized. And also, it's important to have that early interactions with regulatory agencies to have these discussions, because the platform technologies is how they're moving forward with it. They've struggled with all these uniquenesses. You know, what are the rules? What are the regulations? So that now everything's working together. Everyone knows how important these products are, and we need to get them to market. So they go to the platform technologies to understand some of the backbone things that make it easier, and they need to work globally too, to be able to approve the product.
Yeah, there's no one technology that's going to solve all of these problems. It really is going to be, you know, it takes a village to get all these analytical tests done. Everything from, you know, digital droplet PCR for measuring your gene products, and then you got to put these into things like AAVs, and then you have to monitor how many AAVs did I make? Are all my AAVs full? Do I have empty AAVs? And is that going to impact, you know, how much I need to dose with and all the other considerations? So I think as a technology provider, which Gyros is, we're really trying to see from the field, how can we better serve the scientists who need to have robust analytical tools to measure these kind of things. So for Gyros, we're looking at, you know, viral capsid that we can measure, or for biomarkers that can be measured on the patient, and we want to see from the field, what are the best needs? You know, what can we help solve the problems for? That's what we're trying to do.
Yeah, actually for the gene therapy, essentially, that's a system project. One lab can't finish the whole project. We have good collaboration with different labs, try to set up the method, get the reliable and accuracy, and the method that can be used to monitor this whole process. That's why the different tools are used, try to set up the method.
Yeah, and those methods are so valuable once you have them set up, because you can use them preclinically, you can use them clinically, and now we have to do these long-term studies. So they're saying that you'll be using these methods for 20, 30 years, so they're very valuable.
Yeah, especially for the safety monitoring and the immune reactions, that method maybe once the patient get treated with this product, they need the same method to be used to monitor the whole process, because not like the regular molecules, the clears and the drug disappear, they do not monitor anymore. But for the gene therapy, because the new gene going to the patient, it lasts a longer time or even the whole life. That's why you have to use a regulated method to make sure that the method is reliable, reproducible, and sensitive enough, so that they can do the monitoring in the whole process of the treatment.
Yeah, and I think the validations aren't really as black and white as the traditional pharmaceutical methods.
Yeah, definitely, because the technique is different than the traditional, like LCMS, that's maybe not enough for the gene therapy. So the primer probe, such as the digital PCR, you have to do the primer probe, everything, the reaction, the critical reagent, because they all can affect the bioanalytical result. So that's why you have to get the method validated, and just different parameters. Actually, FDA have the guidance for this bioanalysis method. You need to follow this guidance and get the method validated and qualified to support the project at a different stage.
Interesting.
It seems like it takes a lot of collaboration between different teams. If you're using these different platforms, how do you think then we're going to maximize that collaboration and that communication between teams to make sure that, you know, all of these different platforms are being used in the correct way?
It can certainly be challenging in terms of expertise. Some of the platforms require more expertise than others, and then being able to communicate your particular results that you're, you know, very good at to the rest of the team who needs to take that information and build a case beyond that, right? So they're looking at, how many capsids did I produce, or how many copies of gene do I have? And so they need to have that information to build on the next sequence of steps that are going to be taken for the process. And so if you don't have the ability to kind of harmonize that, and I think that's something we're definitely working towards, is harmonizing some of this data to get it cohesive. It can be very challenging without that ability to see eye to eye with your colleagues on what's being produced and what needs to be done next.
I found with some of the cell therapies, because they've taken products that were traditionally for blood banking or at the hospital, and now they're going to be manufacturing GMP product, at least the raw materials. They're not there yet on GMP compliance, and nor are the personnel that are working with the equipment. They don't even know what that is. So I think that's an area for improvement in the field, because you really need that traceability from when the cells are taken from the patient through the whole process.
That's why the regular study is important, because we need a GLP lab to track everything and document all the information inside. So later, when there's some problem, we can always find the root where this problem is and solve the problem. That's the regular study can get this done.
Yeah.
We're seeing a lot of changes and growth in terms of GMP as well for the making of these products, where this is still fairly new for the folks who are doing the manufacturing, right? So it's a lot of work for them to adjust their processes. These are new modalities for them as well, and so some of the things that carry over from biologics or small molecules may not necessarily go one-to-one for some of these new modalities, and so it does take some work on our end to facilitate that kind of communication and transition for them.
I think there was a huge positive and negative with COVID. So COVID showed us that we can get a product to market in 2 years. It also took away from, well, it helped us understand the mRNAs and get that commercial, which is amazing, major platform. But the supplies, they took all the supplies. And so everyone that was working in their R&D, or even the vendors that were supplying the supplies, they were getting strapped and not having enough power to put them out. But it also slowed everyone else down, because they just couldn't get the supplies to do the laboratory work.
Because that's in the urgent need. Everything wrap up all the expertise together, try to facilitate this process to get the product out as soon as possible. Yeah.
And it's largely the same types of equipment. But then I did hear that now there's an excess of the equipment because they don't need to make it all the time. So they're repurposing it back to the gene and cell therapy.
It all comes around eventually, right? Yeah, no, we had global, material shortages for things that you would never consider. Pipette tips and all kinds of stuff were just nowhere to be found at some points. And I think that's shown that there are some serious gaps in terms of what is producible and on short timelines. So we need to, as a supplier of some things, we need to maybe look at our process to ensure that we can meet the demand of the clients, because that's, at the end of the day, what we're here for, is to get you the materials you need to do the work.
So if we look now towards more of a regulatory perspective, what's involved in terms of getting those cell and gene therapies to market?
Well, we're in a very fortunate time where the regulatory agencies do understand the benefits of these products and the benefits they have for patients with unmet medical needs, and they want to help. They want to be involved. They have excellent scientists. They work together. So there's several different regions, the EU, the US, Japan, they all have special departments and programs to help facilitate these products to market faster. So they offer expedited programs. You can start with fast track. They're similar throughout the countries. Breakthrough, once you get that early clinical data. They offer contingent approval, so you can have approval before all of your data is in if you commit to more of those post-approval studies. So you can shave off 2 to 3 years from the standard timelines for drug development.
Actually, the regulatory agency try to, if you provide some plans, they can pre-review and give you suggestions how you to modify your plans and make it more meet the need and meet the regulation requirement.
And which I really appreciate. We're all here for the patients, and we all want safe and effective products. So working together.
They're all so very different in terms of therapies, and so each molecule that's being produced is its own case that needs to be built. And so in a lot of these designs that are being submitted to FDA for these filings, it's really on a case-by-case basis, which is challenging for the regulatory agencies for sure, but also for the scientists, because we're kind of, we like defined methodologies for things, and for some of the cell and gene therapy work, we're still figuring it out as we go, which can be hard for us as we're trying to navigate those waters. So as the field matures, I think we'll see more guidance be kind of built in, but it's a very exciting time because it's very fluid and it's allowing us to really serve those patients who have these, can be debilitating diseases.
Yeah, definitely different gene therapy platform have different advantage and also disadvantages, such as AAV, that's a well-studied platform used for gene delivery, but there's a lot of pre-existing antibody against it. And then neutralizing antibody can neutralize the function and prevent the protein expression in the cells, introduction of the cells into the patient. So almost about 50% of the patient have pre-existing antibody against this virus, AAV virus, which will lead to some challenging part to deliver the gene into the patient and get expressed.
And I think we heard a talk today that post treatment, nearly all will seroconvert at that point as well. So you really don't have opportunities for redosing for these. So we really got to make sure we get it right the first time. So it adds another layer to this design where we have to make sure we get enough product to the location that it's effective and it produces, because we got one shot.
Yeah, and I think another challenge is also because they're typically rare diseases, because you're not manufacturing that much, being able to manufacture small quantities and still being able to afford to do that, or how do you validate it if you're only going to make it 3 times? So that's definitely a challenge, but there's lots of incentives from the regulatory agencies for rare diseases that help incentivize companies to make these products, but also finding the patients, understanding the disease, and understanding what to measure.
So we've talked there a little bit about some of maybe the regulatory challenges with bringing things to market. What about challenges maybe with the actual bioanalytical approaches themselves? Are there challenges there, or do you think this is pretty smooth sailing from now on?
On the support side, so we're getting better, but no, this is still a field that is growing and feeling some growing pains in terms of analytical techniques. We've definitely grown in terms of what we can do. Digital droplet PCR has been very beneficial in terms of getting our genomic workflows done. We've been able to improve some of the efficiency of ligand binding assays for that kind of workflow. But we're not there yet. There's still work to do in terms of making these platforms as robust as possible, because again, they're going to be used for years and years for these ongoing testing. So we need to have a robust platform that's established, will work for 30 years. So work to do.
Definitely, that's why we have the method be validated, because they need to the safety, and they have the accuracy, and also they need to know the sensitivity and the robustness to make sure that the reliable method get it. And not only that, because of the gene therapy, usually the dose is quite low, so the sensitivity is also the challenging part that we need to reproduce the result.
And I think it's hard to understand the PK and the PD for some products. Where do they go? What's the fate of the cells? Are the gene therapies going to a different area? There's so many things that you have to figure out preclinically before you move to the human, but it still is a risk, and it moves very fast. Once you have those early signs of efficacy, everyone's going to push it right on through. So then you have difficulty with manufacturing, because manufacturing's not there. Some contract manufacturers, if it's that platform technology, they've been doing it a lot. There's so many products in clinical research. So those are relatively straightforward. But the cell therapies, if it's not CAR T, it's very difficult to manufacture, and you can have your data, your clinical data, before you're ready to actually commercially distribute it. So it's always, we try to help partner them together.
Yeah, that's why the method they need to be well characterized before the study start. Yeah. So because you need, such as for the cell therapy, they may need to collect the cells from the blood cells from the patient directly, and then modify the genes, and then inject back into the patient for the therapy. So before once the blood is out, you need to complete the process in short time. That's why you have to do the well-cut method. So you need to characterize the cells before the injection and also offer the modification to make sure they are safe and the targeting gene is present over there, go to the right place. Yeah.
One of the things that Gyros is trying to help facilitate is, since a lot of these patient materials are in short supply and the number of patients is so low, we're trying to really minimize the amount of material being consumed, because these gene therapy products are expensive to make, they're expensive to manufacture, and so we're trying to help by really reducing the amount of use per sample test, so we can be accurate, robust, and also not a burden to the patients or to the manufacturing process. So we're trying to help the best we can on that.
Yeah, that's why the MSD method is very critical, because they need a sensitive method. The sentinel rate should be very high enough, so they can be sensitive enough to detect the targeting gene or the information at a very low concentration.
Yeah, because you don't want to have to be doing repeat sample analysis because your analytical technique was not efficient or sufficient to test what you needed to, because that's wasteful and slows us down and wastes material. So we're trying to make it as smooth a process as possible to facilitate that workflow for you guys.
So we've touched on, there are so many different platforms. Cell and gene therapies are such a broad group, really, and we have so many different platforms, so many different methods. Are there any in particular that you think are showing promise that are going to be the primary platforms or methods in the next 5 or 10 years? Or you think, have you got any favorites for the front runners?
Yeah, definitely. AAV, that's the most popular one you choose for gene therapy. Another is that since the COVID-19, the COVID vaccine, now the lipid nanoparticle will be another powerful tool for the gene therapy.
I see mesenchymal coming through. They just have to work a little bit more on understanding what happens in the body, because you transform them outside and you have it characterized, but once they're in the body, they're alive and they're getting more cues, more growth factors, and then what happens? Do they change? Do they go somewhere? Do they die? But there's a lot of opportunities to make different types of cell types. Yeah.
Very exciting.
Yeah, very exciting time.
Yeah. Any, Eric, that you'd put there right in the front?
I'm going to vote again for AV. I think that's currently the front runner. I see it having, definitely a head start for sure, because it seems to be getting, several of them are close or have been approved by agencies. There's a lot of work being done.
There's some breakthrough in the lipid nanoparticle, because with the COVID-19 vaccine successfully developed with this lipid nanoparticle, so they may be powerful tools to use for all the gene therapy delivery.
One of the main benefits of that lipid nanoparticle over doing viral delivery is it's likely to be shielded, so it won't have as much of that immunogenicity effect that we've talked about, where as we use a virus to get genes into patients, it's a virus, and so the body is responding to that and it's generating these anti-drug antibodies, drug in this case being the gene therapy vector, and so we want to find ways to deliver the target material effectively and safely, and lipid nanoparticles might be a sufficient solution to do that, assuming we can get some more work on tropism and targeting and all the other stuff that goes along with it, but it's definitely promising to get to that point.
And so how do you think that the cell and gene therapy approval and regulation is going to adapt then towards those platforms and those methods that we're going to see in the next 5 to 10 years?
I think it's a wonderful opportunity for global harmonization between regulatory agencies around the world. We all want to help patients. Patients are everywhere, they're not just in one country. So if we work together and we approve the methods around the world the same, we all agree. We agree on what's the preclinical, we agree on the clinical, then we can do what we want. What we really are here to do is to get it to the patients faster.
Looking towards the future, what do you think are going to be some of the skills maybe in the next generation of bioanalytical scientists to sort of support these the cell and gene therapy platforms?
I think from the gene part, the digital PCR should be a trend, because that's first is more sensitive. Another is that this digital PCR is no need to standard curve, so they can quantitate directly what the targeting gene is. And for the protein part, I think ligand binding is still will be the powerful tool because it's sensitive and also tests the function in the, of this transgene expressed in the patient.
Yeah, I'd say ligand binding techniques will still be at the forefront for doing that kind of downstream workflow. The other skill set that's probably needed and isn't being well addressed outside of the analytical workflow is actually going to be in the manufacturing side, where we're still a little bit further behind on some of those good manufacturing techniques to make sure that we can produce these products. So I think there's some work to do in terms of making sure the next generation of folks there are able to meet the demands, because I expect the demand is likely to increase, not decrease, for cell and gene therapies.
Yeah, and it's very difficult to manufacture a biological product the same way every single time, and that's what's very important for the GMPs. Something I'm excited about is off-the-shelf technology. So taking away those antigen-presenting proteins that cause GvHD, that cause the rejection of the cells, and being able to have donors that they donate, and then they can expand the cells, they freeze the cells, and then you wait, and then you can even do custom therapies. So you'll take that one and you'll add on what you need, your genetic thing to go to help with your specific rare disease, rare condition, and have that customized treatment option. When I was looking this up a little bit, it was saying that you have to look at the transgene, but you also have to look at the vector. So you have to do twice as much analysis.
Actually, for the analysis, we talked different levels. First is the delivery vehicle, like using the AAV, you need to monitor the AAV used by analysis. Another is that for the transgene proteins, because when the gene going to the patient, you need to detect what's the gene. Is there gene over there or not? And the third is that did the gene go to the right place? That they call the distribution study. If they want to go to the liver, did they go to the liver or go to other organs? And the third part is for the protein expression. Because you deliver the gene into the patient, you need to make sure that the right protein is expressed. And the proteins function well. And the fourth part is the immune reaction. Because the patient previously do not have these proteins in their bodies. And now you insert the gene and get the protein function well. Now you make sure how the immune reaction will recognize this protein and get the immune reaction. That's the 4 different levels of the bioanalysis we did.
And then that's really interesting. And then would it be different per product? Would you be doing biopsies, or maybe some it would just be a blood sample? Would it have to be different types of samples?
No, that's actually for different methods, different analysis, you use the different tools to do analysis. Such as for this gene proteins, you have to use gene levels. You have to use the digital PCR or the qPCR try to quantitate the gene. But for the proteins, you use the ligand binding, and also for some of the protein function, you have to use the cell-based assay maybe or the flow cytometry to characterize the protein expression or the function on the cell surface. And also for the immune reaction, that's different. You have ligand binding. You also have the cell-based assay, try to neutralizing assay, try to characterize the immune reaction from the patient. But most of the time, you have to use either collect the blood or the cells from the patient to do this characterization.
And the other thing that you can also consider when you're looking for outcomes is you can look at surrogate biomarkers for did my gene product have an effect, right? Am I getting the right kind of restoration of function or things like that? So you have surrogate markers that are also important for doing that, and that's also typically ligand binding assays are pretty efficient for that as well. So there's a number of different modalities that go into ensuring that we got the gene where it's supposed to go, it did what it's supposed to do, and it's safe, right? And so it's a very complicated analytical workflow, and that's why it's so important for us to get these validated methods and get them off the ground.
Yeah, that's what we call the biomarker analysis, right? In different tools, and some of the, even for this, most of the cytokines, even you can use commercially available kit for the analysis, but then make the method more reliable because they are commercially available and easier to do the quality of this kit control, method control.
I'm also very impressed with next generation sequencing, where you can use it as a companion diagnostic. So you can see you have circulating tumor cells in your blood, and it's decreasing that burden on the patient. So a biopsy is a huge burden on a patient. So being able to sequence your cancer cell from your blood, even if it's a solid tumor, and then being able to find that treatment that's been known to work for it is also an emerging field that's even brought back solid oral with great results.
Yeah, that's why this new technology need to appear to use applied for the treatment. So for the monitoring, for the bioanalysis, definitely the next generation sequencing will be used for monitoring some critical factors in the therapy process.
Yeah, very exciting.
Closer and closer to personalized medicine. We're not there yet again, but as we discover more and as we get to the opportunity to really tailor these treatments, that's another application where the cell and gene therapy field has real opportunity for growth in terms of really efficiently treating these diseases.
Yes, because I think with small molecule and large molecule, they're doing the one treatment fits all. You have this disorder, you will get this, and that's not, we know it's far more complex now that we can sequence these things and truly understand what the problem is.
Definitely, because previously for the small molecules, you need to multi-dose, and also you need to check it, and once the treatment stopped, and then disappeared, no anymore. But for the gene therapy, you actually insert the gene into the patient, it grows, and it have a longer safety effect. That's why the bioanalysis is more important, because you need to not just monitor at the beginning, but the long term, the whole process, maybe the whole life, because to make sure that there's no any side effect to appear, and especially when they use the lentivirus to deliver the gene into the cells and then inject the cells back into the patient.
Yeah. Another interesting thing with the rare diseases, you have to treat as early as possible, and it's very difficult to know there's a problem. It's a baby, and you notice maybe something's a little off, and so they're going to go through all these tests, and time is going by. So being able to get those early detections, and then you give that treatment before, because the way you develop as a human being, it's everything relies on each other, and it's a very beautiful thing. But with the genetic disorder, something is off, and it starts to spiral. So the earlier you can get in to help, and the earlier you can detect, will also help move this field forward. But then it's all unique. It's all unique, because so CAR T, it's a cancer you develop later. So every single different thing you're trying to treat is very unique.
I think that shows the breadth of this field. It has the opportunity to treat a wide variety of things. The challenge is it has the opportunity to treat a wide variety of things, right? And so there's so many analytical challenges that go with that, and manufacturing challenges that go with that, and regulatory challenges that go with that. So as we come together and form consensus here, I think that's really key for us to drive the field forward.
And maybe that'll change industry. Big Pharma has always been so about the money and then blocking biosimilars, and I just don't feel like it's always been for the patient. But maybe we change that. Maybe we work together to get things faster, like it is in academics. We always work together. We shared ideas. All of a sudden, I'm in industry, and it's like, no, but it's for the patient. So we need to come back to that. And I think COVID, we learned to do that. We work together.
Yeah, like COVID-19 vaccine, they
Yes.
developed in such a short time, and every part work together, from the manufacture part, for the monitoring part, and even for the bioanalysis techniques to work together to bring all the resources, and it will definitely foster the process of this development.
I know, I mean, it was a huge negative with COVID, but that's the huge positive, to show that we can do that, we can work together.
Wow, that's really exciting. Well, thank you all so much for joining me on the panel. It's been really lovely to talk to you all.
Happy to be here.